All right, you guys, welcome to molecular biology, and we are going to split this chapter into two lectures, okay? So the first lecture, we're going to really talk about the structure of DNA. So we talked a little bit about DNA when we were talking about the different nucleic acids and things like that, but we're really going to get into how DNA is structured and why the structure of DNA is so important. And then we're going to discuss DNA replication today. So how do we actually make a copy of our DNA during the cell cycle? So remember, we have interphase where we're going to be resting and getting energy in the cell, but then we also are going to replicate our DNA to get ready for cellular division, whether that's mitosis, usually, or meiosis. So who discovered or determined the structure of DNA? Well, Francis Crick and James Watson are always, credited for determining the structure of DNA at Cambridge University in England during about the 1950s. But there's also some other people that are highly involved in how to image DNA or how do we look at DNA. And so Linus Pauling and Maurice Wilkins were also kind of exploring the field of DNA, and they were using a method called X-ray crystallography. Now, Rosalind Franklin became famous as well because she actually used this technology to create an image of DNA. And so in 1962, of course, Watson, Crick, and Wilkins were the ones to receive the Nobel Prize, even though Pauling and Franklin were fairly well involved as well and were famous in their own right. So what is the structure of DNA? We've got two types of nucleic acids. If we go back to our talk about organic molecules, right? We talked about deoxyribonucleic acid, which is DNA, and ribonucleic acid, which is RNA. And the building blocks of DNA are those nucleotides. So we talked about nucleotides, being four different bases, right? Named after their different bases. So we'll talk a little bit more about that. So the nucleotide is going to contain three main groups. So we're going to have a sugar, right? We're going to have a 5-carbon sugar, and that's either going to be deoxyribose or ribose, just depending on if we're talking about DNA or RNA. Then we're going to have a phosphate group. Okay, so the phosphate group is always going to be consistent. And then we have a nitrogenous base, and that's what the nucleotide is named after, is the base. So the bases are going to be adenine, which we term A, guanine as G, cytosine as C, and thymine as T. Okay, so we have two different types of bases, and these are our pyrimidines and purines. And so don't worry too much about that, but know that there are two kind of main types of bases, right? And so that's going to help determine who is going to pair up. There's two main types of bonds in how we actually build our DNA. Okay, so what is going to bind to what, essentially? So first, we have the phosphate groups are going to bind covalently with the sugar molecule, so with the deoxyribose. Okay, so we have the sugar phosphate backbone, as we call it. So the phosphate is going to covalently bond to that sugar. Okay. So then we have a long polymer of these nucleotide monomers, and this provides the backbone of the DNA. So if we keep repeating this, sugar phosphate backbone and they keep binding over and over and over again, we're going to have a long polymer of these nucleotides. So remember, monomers are just the single nucleotides, right? Single nucleotide, and then we put them together and we create a polymer. And so this is really the structural backbone of DNA, okay, is this sugar phosphate backbone. So once we have that backbone built, then we have a bunch of bases that are going to bond together, and they're going to use hydrogen bonds to connect the bases. And so essentially we're going to have two opposite strands, right? So they're each going to have a backbone, and they're going to be running in opposite directions. And then their bases are going to pair up. So we always say that the same bases pair to each other, right? So adenine is always going to pair with thymine, and cytosine is always going to pair with guanine. And so you're always going to have a pyrimidine base paired with a purine base. Okay, so that's kind of where those different structures of the bases come into play, but don't worry as much about that. Just know which bases are going to pair with each other. Okay, so the oxygens and the hydrogens and the nitrogens and the hydrogens are going to create this hydrogen bond between the bases. So it was Watson and Crick that proposed the idea that DNA is made up of two strands kind of twisting together. And so they came up with the double helix idea. And so it's really two anti-parallel strands. So essentially, if you say that there's a direction of each of these strands, right? And it depends on the carbon direction. So don't worry too much about that, but they call it the 3' end and the 5' end. And it just depends on the direction of the carbon or the position. And so if you have one going in the kind of upward position, 5' to 3', that one's going kind of left to right, you can think of it. And then the other one is going in the downward position, so you kind of have the 5 to 3 going downward in this picture, right? You can turn that horizontally, you can change it, but essentially the idea is, right, that you have two different ends of the strand, and they're going to be going in opposite directions, right? However you want to think of that. So we're not too worried right now about what 5' and 3' means. It just means that they're going in opposite directions. So if one is going left to right, the other one is going right to left. Okay. So here's just kind of a picture of a ribbon model or image, kind of a chemical structure model, as well as a space filling model to kind of show you how DNA can be depicted. Okay, so on the left-hand side and the right-hand side, you're getting an idea of that double helix and the rotation of DNA. Okay, so you can kind of see how it rotates together, how it spirals together. And then in the middle picture, you can see some of these bonds and the differences between maybe the ends, the 5' versus the 3' ends, okay, and how those bases bond together with the hydrogen bonds as well. Okay, so just kind of a couple different types of images on how you can view DNA. So now let's talk a little bit about RNA, which is our other nucleic acid. So instead of deoxyribose, we have ribose. So it's still a polymer of nucleotides, but it has a couple of differences. And the biggest one is that it is single-stranded instead of double-stranded. Okay, so it only has a single strand of nucleotides. It's not double, so we don't have the base pairing like we do in the double helix. We also have the sugar as ribose instead of deoxyribose. Okay, so that's a little bit different. And then when we do have the bases, so we replace our thiamine base with uracil. Okay, so it's uracil instead of thiamine. Okay, so when we say that adenine and thiamine always connect, when we are connecting RNA bases, it's going to be adenine and uracil instead of thiamine. Okay, so wherever you see thiamine, we're going to replace it with a uracil. Okay. So what about DNA in the cell? So we have all of this DNA. It's really, really long, right? So if we have all these strands of DNA, double helixes, it's about 6.5 feet of DNA. So how do we fit 6.5 feet of DNA in a microscopic cell, right? So how does that happen? Well, we package it in a very specific way so that we can fit it into the cell and we can still access it and have it function correctly as DNA. So we can still read it, we can still replicate it, but it's packaged in a very specific way. And again, we have to have access to this DNA for not only replication, but also transcription and translation. Right? So we have to package it very specifically so that we can still have access to it, but it's going to fit into these microscopic cells when we're 6.5 feet long. So how do we package DNA in eukaryotic cells? Well, the DNA is going to be wrapped around these proteins called histones. And once they're wrapped around the histones, we call them nucleosomes. Okay? And then these nucleosomes are going to coil. Okay? So they're going to kind of create their own helical structure, and we call that a chromatin fiber. Okay? So that is going to create a spiraling effect, which we then can package even further. So then we can then condense these spirals into a chromosome. So if we're able to continue to spiral and even supercoil these fibers, then we can package them into chromosomes. Okay. So I have a great video on how DNA is packaged. So I've posted it on Canvas, but you can also follow the link if you have the PowerPoint up. And it just really gives you a great kind of video image of how we package DNA and how the supercoiling actually happens. Okay. So on the right hand side, we have some picture depiction of what's happening. So we know our double helix, and then we wrap that double helix around a histone. And a histone is just a protein. Okay, so the protein, remember, has its own specific folding pattern. So we have some alpha helices and some beta sheets, right? So that's what it looks like if we wrap it around a protein. And then those nucleosomes, so once we wrap it around, we call it a nucleosome, they're going to coil into chromatin fibers. And then that's going to further condense by supercoiling, okay? And then that chromatin will then be part of a chromosome, okay? So what about prokaryotic cells? So even though their DNA is quite a lot shorter, it's only 1.6 millimeters long, it still needs to fit into a very, very small cell, right? Prokaryotic cells are often quite a lot smaller than eukaryotic cells. And so they have a circular structure of DNA. It's still double helix structure. And what happens is that ring essentially supercoils. So I don't know if you've ever taken a string and kind of twisted it, and then you can see how it coils. And that's essentially what's happening to the DNA. And so in the there's a picture representation as well as an electron microscope picture. And so then the cell is able to package it a little smaller into the cell. Okay. So go ahead and pause the video. We'll do a little bit more review. And so let's answer these questions. What are our 4 nucleotides in DNA? And what are the 3 differences between DNA and RNA? Okay, so let's go through the answers. So our 4 nucleotides, remember they're named after the nitrogenous base. So we have adenine, which pairs with thymine. We have guanine, which pairs with cytosine. Okay. And then what are our 3 differences between DNA and RNA? Well, the D and the R is very obvious, right? So D stands for deoxyribose and R stands for ribose. So we have a different sugar. So they're similar. They're both 5-carbon sugars, but RNA has ribose instead of deoxyribose. And then another thing is RNA is single-stranded, so it doesn't have the double helix or the double-stranded structure like DNA does, okay? And then we've also replaced one of our nucleotides or one of our bases with uracil instead of thiamine. So adenine is going to pair with uracil instead of thymine in RNA. So what is the big point of DNA, right? Everybody talks about how important DNA is in the cell. So DNA actually has 2 big functions in the cell, right? So first, and the one we're going to talk about today, is DNA replication. So we're going to copy our DNA so that we can undergo cellular division. Now this is very important for growth, repair, and reproduction, right? So the other big function is that it undergoes transcription and translation. So essentially our cells are going to read the DNA and then translate it into a protein molecule. So this is how we build proteins in our cells and in our body. And then those proteins are going to be very important for different cellular functions and body functions. Okay. So we'll talk more about transcription and translation in the next lecture. So we're going to talk about DNA replication today. So this is when a cell divides, you're going to get an identical copy of the DNA in each identical daughter cell, right? So how do we make a copy? Right? So how do we make a copy during interphase for cellular division? And that cellular division can either be mitosis or meiosis. We still have to replicate our DNA first, okay? So this just gives you a basic idea down here, this schematic, is first we have to unwind the DNA, right? So the base pairs have to come apart, and then essentially we're going to copy each side of the DNA, each strand, okay? And then we're going to make a copy from that strand, so then you have another double helix, right? So then if we make a copy of each of those sides, then you've doubled your DNA, right? You've replicated your DNA, you've made an identical copy. Okay, so this gives you a good idea of how this kind of works, and then we're going to go through the specifics. So I have a really good DNA replication video if you're interested, and I highly recommend watching it because I think videos can definitely give you a great idea of what is actually happening. Because essentially we have to kind of unzip the DNA and therefore separate the DNA so that each strand becomes a template for a new strand. So essentially, we have to copy each strand at the same time. So remember, they're going in different directions. So essentially we have one leading strand that's going to be copying in the direction of the DNA. So it's very easy. But then we also have a lagging strand, which essentially is going the opposite direction. So that is kind of the complicated one. And what is doing the copying is an enzyme called DNA polymerase. So it is a protein, an enzyme that essentially is going to be creating these new strands from the old strands. So creating templates from the old strands. There's also another enzyme called helicase that essentially is what is going to be unzipping the DNA as it goes. Okay, so again, if you kind of watch the video, hopefully this will help to solidify kind of what's going on and what's happening. But we'll go through the steps as well in the lecture. So the first step of DNA replication is called initiation. So essentially this is where we have to unwind or unzip the DNA. And we call this the origin of replication, and we use that enzyme called helicase. Now remember, we have so much DNA, right? 6.5 feet of DNA. There's no way you're going to find the very beginning and the very end. So essentially you're gonna just have to start in the middle, okay? So essentially that means you're going to have to unzip, kind of going in both directions. So we have two things called replication forks at either end. So you're going to kind of open it right in the middle, which is the origin of replication, and then you're going to unzip in either direction using that helicase enzyme. Okay, and you can have multiple origins of replication when we're talking about eukaryotic chromosomes because there's just so much DNA. So this is going on in multiple areas of the chromosome. Okay, so we have so much DNA to replicate that it's going to take a lot of work, right? So then the second step of DNA replication is elongation. So we have to add new bases to that parent strand using that enzyme DNA polymerase. And we actually start with an RNA primer. So that kind of tells us there's our starting spot. Okay, and then essentially using that DNA polymerase, it's going to start adding on those complementary bases using that parent strand as a template, right? So it'll be the opposite base of what is on the original DNA. Okay. So the leading strand, essentially we said we have to have kind of a leading strand and a lagging strand. So this is made continuously because it's in the correct direction, right? So say in our picture here, that top strand is going in the 5' to 3' direction, or left to right. That means we're going to make a copy right to left, and that's in the correct order, right? That's how that other strand would have been oriented, and that's the leading strand. It makes sense, it's going in the right direction. You can just continuously lay down the bases and not have a problem. But the lagging strand is the challenging part. So the lagging strand has to be made in pieces. So remember, so now we're going right to left, right? So now we have to actually recreate it left to right, which is the opposite of what we were doing originally. So essentially we have to lay down these fragments, and they're called Okazaki fragments. And then essentially later we'll have to string it all together. Okay, so the DNA polymerase can really only go in one direction. So that's why we have to create these fragments and then string it all together later. Okay. So once we have elongated the DNA as long as we need it to be, we have what's called termination. So eventually that DNA polymerase is going to come to a repeating sequence of DNA. And this repeating sequence is called a telomere. And essentially it's the end of the chromosome. So it's saying, okay, you've finally reached the end, you're done, you can stop. So essentially once it recognizes this repeating sequence, then the DNA replication has stopped. But we have to go and fix a couple of things. So we have to remove those RNA primers and add new DNA nucleotides in the place of the primers. And then we have to seal the backbone and our fragments with another enzyme called DNA ligase. So if you think of DNA ligase, it's kind of like the glue that puts everything together. So it's going to put it all together, take the primers out, put in the right nucleotides, and then we're good, right? So at the end of DNA replication, you have an exact copy of your DNA, right? So you're using those parent strands to create the opposite parent strand, right? So then you have two exact copies of your DNA. Now, obviously, there can be some problems that can happen during DNA replication, especially with that lagging strand. You're doing it in fragments, so there are errors that can happen, which is why we have checkpoints in the cells to be able to make sure we have undergone DNA replication appropriately. But that's also what gives rise to mutations too. So we have mutations that either, don't do anything or can be harmful or are just neutral or even positive, right? So sometimes mutations can be positive and lead to, changes in evolution, but this is where it can happen because it's not a perfect model, right? So lots of things can happen, and I highly recommend watching the video to really show you how DNA polymerase works and functions and how it's actually bringing in those bases and pairing it with the parent strand. Okay, so let's finish off with some review. I know this was a shorter lecture, but the material is quite complex. So I want you guys to review and make sure you understand the steps and don't get too bogged down with all of the details. I just want you to understand the steps and what's happening during those steps, as well as what the end product of DNA replication is. So go ahead and pause the video and answer these questions. So what are our 3 steps of DNA replication? And what is the end product? As well as what makes it? So our 3 steps of DNA replication are initiation, right? So we have to unzip the DNA. We have elongation, right? So we have to copy it, copy each of the parent template strands. And then we have to terminate it, so termination. So initiation, elongation, and termination, okay? And then we end up with an identical copy of DNA. And it's DNA polymerase that is making those copies from the template strands, okay? So if you have any questions, definitely let me know, because I know this process can be complicated and complex. So go through the videos, make sure you understand what's going on during DNA replication, okay? And I'll see you guys in office hours. Thanks a lot.